today we are going to talk, I'm going to talk a little bit about the aminoacyl-tRNA synthetase, whose main function is to the deciphering of the genetic code. And so these are very important enzymes. These enzymes, they charge the amino acid on the terminal end of the transfer RNA. They charge normally when the charged tRNA reaches the ribosome. That's where the amino acid is here on the 3'. But I'm going to present shortly a brief history of these synthetases, just to introduce how the structure became a crucial partner of our understanding. So it all started in the 58, well, a little bit. I mean, 58 was the presentation by Francis Crick. He had what is known as the adapter hypothesis is proposed. That was after the discovery of the structure of DNA matching. And so a major question was, what is the genetic code? Is there a genetic code? How much does it represent? How is it translated? How do we go from DNA to protein? This is one step. And Francis Crick was a visionary. He made the hypothesis that the faithful synthesis of proteins from messenger RNA is dependent on the presence of perfectly acylated tRNAs. Okay, that's, you can say, a trivial observation, but the hypothesis suggested that each aminoacyl-tRNA would be made by a unique enzyme. This is a key question. This is the adapter hypothesis. So it is one enzyme per amino acid, and as many tRNAs as possible. But then the question at the beginning is, why is that so? So you read the paper, you will understand the theory behind. It's quite simple. I mean, the system would not work, actually, with total confusion with a pool of synthetases. It's not viable. But how the synthetase charge various tRNA. I raised already the question previously. And then the question is, how do they work? So there are two steps. I will not go into too much in the chemistry of that. There are two steps of in the reaction, aminoacylation reaction. First, you have ATP and the amino acid. They form the adenylate. So that's the first step of the reaction. The second step is that the adenylate reacts with the transfer RNA, the CC end, and the terminal adenosine, and charge. Then that's the second reaction within the same active site, charge the amino acid to give that what I showed before. So just a little bit of history and how I became involved. Why am I here to talk about that? You know, in the 74, I was a young postdoc with Michael Rossman and chemist training. So for me, molecular biology was 1 year old when that paper came out. I joined in 73, and to do structural biology. So, and well, I was not working, I was working on GPDH. But we found, see, I say we because I was part of the team. Michael was the head and the mentor behind. Found that there was a domain, a molecular domain with a specific fold that was found in different proteins, a lot of different proteins. And we tried to figure out which function they had. They had a common function. At least that's what we picked up. They would bind nucleotides. Nucleotides at that point were coenzymes first. So they were binding NAD, NADP, ATP, kinases belong. And the structural biology in that here, you know, at that time was not so great. So there were not so many structures, but a lot of them had that. And so we came out by comparison, by structural comparison, because the sequence analysis would not have given the solution at that point. That there was a three-dimensional alignment, the fold, that had a function, so it was a domain and function. And we had some measure at that time. That's a primitive evolutionary approach. I mean, these changes that suggested that the structure was present during pre-cellular evolution. So it's very old. It had to be a very old protein because there was a very primitive function. So that was the assumption. And actually, if you read the paper, you see at the end, among the prediction of a molecule that would have this fold were aminoacyl-tRNA synthetases. We had no structure at that time. The first structure came, you see, 15 years after. And that was the first structure that was done in the group of Tom Steitz at Yale, working with Dieter Seul, who is one of the pioneers, also the work with Paul Schimmel of this group of synthetases, of this group of enzymes, sorry. And the idea, the structure, for the first time, we could see the structure of the protein bound to the tRNA. And indeed, the molecule, the active site of the molecule had by the time, what became the Rossmann fold. So happy, we have the solution. A very old protein, enzyme, stick together, and so evolution is very simple. We have the very old model protein, and that's where it comes from. And everybody was working hard. It's a deep question, try to compare all the enzymes. But in the meantime, all sequence analysis, primary sequence that had been with more data, could never give a proper alignment of these enzymes. Never. Well, it was a big mystery. And so the structure was correct. And actually, if you read this first paper, it says, all the conclusions. And one of the basic conclusions is that now we have the active site, and everything is evolving from that. So the conclusions were based on the assumed fact that the family was a family of enzymes with one active site and domains. Makes sense? Everything made sense. Makes sense few months. Because that's where we came. My group was published in 1990, but the discovery, is something very close. Appeared, I say, few months, with two very smart guys, Marc Delarue and Olivier Poc, who were very smart in terms of sequence analysis. They use very sophisticated approach, not only the classical one, looking not only for homology, but also a very important thing. If some residue is very important, not only should it be conserved, but if you introduce it somewhere else, it has to be exclusive. There is a double condition. Importance of the residue, and it cannot be put in another place because it will destroy. So by combining that, found the alignment. And if you go to the paper, it was very few amino acid conserved. But the fact is the synthetase could be partitioned in two classes, two families of enzymes. And there is a story also. You see, you have these domain had a lot of people involved, and chemists were involved. And two groups of chemists, but essentially the group of Kramer in Göttingen, had tried to address, were addressing questions from all sorts of facts. I told you that it was accepted. It is accepted, actually, it's known, but it was accepted that the hydroxyl group was the place where the amino acid was attached. But as a biochemist, one would think, there are two hydroxyl. Which one is the target? Where is it attached? People didn't know exactly. So that was a question. They had done a lot of sophisticated work, and they had found, that some had attached in 2', some in 3', So there was a table like that, where distribution and no logical correlation, which is strange. So but the work was so important that you had this table. And by chance, having in mind the partition, I was looking at the presentation where this classical table of genetic code, every talk had the genetic code, with the 2' and 3' hydroxyl group pointed out to each corresponding synthetases. It struck me that, boy, but that's the correlation. One class is charging 3', and the other one is classing 2'. And then, of course, that's the luck to be chemist. For a chemist, you have two enzymes, so the target is different. What is different between the 2' and 3'? The difference is that there are two different chiral centers. The chirality is not the same. There are two different chiral groups. Then it's a basic rule of biochemistry, chemistry. With an enzyme, you cannot charge two different chiral centers. Chirality is a very deep difference. So that was the chemical explanation, and this was. And then soon after, we had the structure of the class 2 with the transfer RNA. And then so since we're speeding, now summary, these are the two classes. Say roughly the same number of amino acids. That's another question. I will not want to. The very obvious observations sometimes raise the most important questions that nobody could answer. I mean, they still why do we have the same number of amino acid group partition in both? I will not address this question, especially Silvano cut me out in time, so I cannot speak about that. It's not true. But there are mechanisms. It's not proven. But you see here is the Rossmann fold, and that's the other fold. What is interesting from the evolutionary point of view, I pointed out to you that this is a very common fold. All kinases, it's one of the most abundant domain found in the protein structures. The class 2 active fold is very rare. Very few have. It is interesting for what has to be one of the oldest enzymes, that it has not been used elsewhere so much. Well, that's, and I'm trying just to see how think in terms of evolution. One possibility also, since you have that system highly conserved, it would, and the other one is spread all over, is that in fact they didn't start together. And then one group of enzyme started before the other. But that's too long for the discussion. There are several points. Now let's go just observations. So we have these two. Oh yes, another point of interest. When you look, I told you 2' and 3'. Every rule has exceptions. If you have a rule without exception, you wonder, what's wrong. And in fact, there are two enzymes where you have 2', 3'. That does, I told you, the chirality is an essential virtue, so cannot be one or the other. So it just means that the control are not as difficult to make. And here we have phenylalanine, which is a 2'. I'm still wondering if that is correct, but it's there. The other point, which is a bit more interesting, I will address today. Here they are essentially monomers, the functional, except these two last, which are dimers. When I say monomer and dimers, that means this is a feature which is conserved throughout species, it's a very dominant feature. Because you can have oligomers formed in specific conditions. These are not really constrained functional oligomers. What is an important thing is that these are all dimers. We'll see now if dimers, the class 2, are obligate dimers or not. So if that is a constraint and it has some functional correlation, exceptions must be explained. So we're going to address that question today. Just a little bit of history. Is here the active site? That's what published a bit later we did. It's a stereo view. It's just to give you an idea. In 20 years, a bit more, 25 years, how I already pointed out 2 days ago, how the quality of the information is moving. But this, if you look at the stereo view, the conformation, the density is better. This is a stereo. If you can look at that, you have a 3-dimension. What you have here is the ATP, and here is the adenylate. What is important, an important feature, is that the structure of ATP in the class 2 enzymes is really non-typical. Actually, it's, to my knowledge, close to unique. All ATP binding enzymes, like kinases, other things, they have an elongated shape with something more similar to that one. And this is, you see, it's a U. It's a very sharp turn. And this conformation is necessary for the mechanism. Why is that? It's an important question, stereochemically. Why is that maintained? It's part of the mechanism. And why is that, to my knowledge, it's not found elsewhere? So for an important feature of cell life, and that's being done for alpha things, it's a strange correlation. But that's a fact. But there is no exception to that rule for the class 2. What does that mean? The 2' and 3'. If you now, this is a result of the structure. We compare the structure. Here you have the adenylate, and that's the tRNA CCA end. The fact that the structures are different, the binding of the tRNA is different, means that the CCA is coming from opposite side. Or another way to put it, it's not the tRNA which comes from opposite side, it's the substrate to which the tRNA will attach that is positioned differently. So in the class 2 here, it comes from this side. In class 1, they come from this side. The protein is bound here. The protein is here. The CCA, in this case, the protein is here. In all these comparisons, it looks like if we add these enzymes going through a mirror, you always have a repetition. Always you find that duplication. Of course, that's also something, a good take-home lesson for you starting a career of scientist. If a result is for an important question, and what is more important than the translation of genetic code, or well, there are other important, but that's a key one. Regulation, for example, of gene expression, tackle cancer problem, what is the real starting cause of what. Then after that, the cascade is nice, but the key question at the beginning. When you have questions like that, if someone comes with an answer, it will be tested. If nobody is interested, then okay, who cares? But this question of the 2 classes was really bothering people. I mean, are these 2 classes unique? Because once you have, go from one class of enzyme doing a job, then you find, no, there is not one, there is 2. Well, there can be more than that. So people were chasing. And one of these ideas, to the exceptions, is that there was a mystery. Some archaea, I think I will come back to that. Yeah, I think it was an archaea. One aminoacyl-tRNA synthetase is missing. Then if you have, this is the cysteine. Cysteinyl is missing. So you'll find the whole gene, there is no cysteinyl tRNA synthetase. So what happens? How does it work? It cannot be, it has to be gone. Well, I leave aside the hypothesis, we have now the solution actually, but one result which was very provocative, appearing, that's about 10 years later, the two classes in Science. So, it's not because it's a big journal that it is true. And that's also another take-home lesson. And that, so they found the observation through not structured genetics experiments. They found that archaeal pro-cysteine tRNA synthetase contradicts the adapter hypothesis. That's a TIPS paper. Where people explain, okay, Crick was wrong, or partially. We have one synthetase who charges both tRNA, different tRNAs. So two substrates, two tRNAs in the isoacceptor with one enzyme, one catalytic site. That's a miracle. And to tell you the truth, I was quite negative. I mean, to try about that. You can understand how structural biology didn't make sense. When you see things like that, you say it's not the poor Crick who is contradicted, it's chemistry and physics. Okay, one synthetase, talk. Just to give you, when you have such provocative ideas, that would have been another revolution. Then for 5 years, there were lots of papers in the top journals proving that, no one disproving actually, trying to show how it works. A few years. And turned out the explanation came a bit later. First, the observation was due to an artifact. But then we could understand, and it came another paper by partly the author of the first paper, and with a group of Tom Steitz, he helped them to do the structure of the complex, and shows that this is just, I would say, you can read the paper, it's a minor component, it's an artifact, okay, to summarize. I mean, they can explain the artifact. So the explanation came, they could explain why the observation was made, was an artifact, and back to the owner. Another example, is something which appears all the same times, 10 years later. 2001, Nature, before it said Science. Aminoacyl-tRNA synthetase whose sequence fits into neither of two known classes. Bingo. There is a third class. This paper was never withdrawn, but this was not a synthetase. It was in Nature. That's also another take-home lesson. Not because it is in Nature that it is true, but normally it is most of the time, isn't it? Okay, the discussion yesterday prompted me to add a little bit more on that. But now, about the explanation, I showed you already that if you take in terms of evolution, seeing is believing. You present, this is usually the way you see the genetic code presentation. If you just change the order of sequence, it's the same genetic code, nothing changed, except just the distribution. You see how the color is distributed, and you have class 1, class 2. Well, that tells you that, for the cytosine in the second position is class selective. So you start to have some hints now in terms of evolution. But the problem, this is good to see the evolution of the genetic code and correlation. The problem is, I told you Tuesday, that the anticodon, which bears the genetic information on the tRNA, is not the feature which is always recognized by the synthetase. So it's a bit more complex. But it is true. This observation is true. So because there is a correlation between the enzymes and the genetic code, of course. There are genomic surprises. Some of them are here. Absent, some synthetase are absent, present, or absent in archaea. This was another puzzling observation now that we have the full genomes. In some organisms, it's absent. So the mechanism is using bypass. And this has been now investigated, notably the group of Dieter Searle is doing a lot on that. And so they have also done structured collaboration with a group of Nureki in Japan. And this is one example of an archaeal non-discriminating glutamyl-tRNA synthetase, a missing link in the evolution, where you go through one synthetase and then modify in order to get the right transfer. So this is, we are still in the field, but you see how nature can bypass some problems by adding some complex modules. You know, I have no time, thanks to, you know, to go too deep. But these enzymes, there is a primitive startup. The active site, I will come back to that. But then now they have evolved into, these enzymes are working in a more sophisticated cellular world with different cells. So there is an adaptation. So for the early beginning, we concentrate on these highly conserved parts. For today's function of these enzymes, even if it is in translational genetic code, they have to adapt and they acquire more sophisticated structure and function. So these are the two aspects of the evolution. So if you are interested in really the early basic question, this is the question I raised before. If you are interested in the evolution of these enzymes to say today, you have to go to these more sophisticated and specific. Then it becomes a question which can be used, because what's the interest of studying these aspects? Is that because you have cellular differentiation? Why is it interesting to work on some mitochondrial tRNA synthetase? Because these are really good targets to cure some disease. But the main problem, they are so essential that the targets, if they are really useful, they can be very toxic for the drug. So toxicity is a major problem. So we have to find ways to target these enzymes in a way where you don't touch the basic function in human cells. You see? So this evolution of the structure is important for medical application, not only for basic science. So I just very quickly now, because they are modular, these enzymes. I told you this is the conserved core. So there are two domains which have the active site, and then the synthetase are built by adding other domains. So each domain a function. Here, this domain has a function to recognize the anticodon binding domain. For this enzyme, the anticodon recognition is important, so you have that. But you see, even for the same enzyme aspartic, if you go from eukaryote to prokaryote, that's E. coli, you have this extra module, which is different. So for the same enzyme, different species, you can have very important differences. Right. So the functional correlation are there. Okay, this is the same where that's the structure of the complex, how it looks like. You know, here are the active site, here the anticodon. These are the key domain. This domain has a different function. I have no time to go in. Another function important for translation of genetic code is the problem to correctly discriminate between amino acids which have very close chemical properties. This is called the proofreading. In this case, you see this amino acid, for example, if you take isoleucine and valine, the side chain is only differing by one methyl group, which means that if you have an active site of isoleucine, which can accommodate this large one, it will accommodate the smaller one very easily. So there is an ambiguity in selection. Then there is a mischarging. And the mischarging rate at that level is about 1/200, which is far too much. That would be, it's very, so it has to go at least 1 to 10,000. For this specific enzyme, then nature found a proofreading mechanism. The other case is this one, the threonyl, which is one of the most sophisticated. Serine, threonine, valine, these three can be charged by threonine, and if there wouldn't be correction, these enzymes were, the translation and the protein synthesis would fail, because after that, at the ribosome, there is no control at the peptide, at the amino acid level. And look at the place, the threonyl-tRNA synthetase, which was solved in our lab already 15 years ago. And you see, it really looks like, see, that's what I showed you before. This is the aspartic from E. coli. Look at the threonyl-tRNA synthetase. You find the same basic, this is the active site, the same dimeric, everything is the same, but you have this module, which here is doing other things. That's the module which correct for mischarging. One question I will address. Are class 2 amino acid RNAs obligate dimers? This is something I told you. There are dimers. We don't know what. So one thing, first, we have now more than 400 structures. Physics structure in the PDB. So you don't need to do that. You're just a computer, a PC. We're talking about what we are doing. A PC and some knowledge, you superpose. I had a good student, and Brice, and he did that. And so he analyzed all the structures. I take just for the class 2. This is one subgroup. These are the active sites. What is important, you have to notice, you have the two colors. In red, you have the two active sites, dimers, 1 and 1, and this yellow part, which is strictly conserved, which links the two active sites, and highly conserved. So it's a conserved feature in sequence and structure. View down the axis. 5 others, glycine, proline. And you see the other domain can be quite variable, but you have always that with a yellow connector. So with that we have 8, actually 9, because lysine 2. We have, yes, let's say 8 amino acid of class 2, same structure, the dimer, everything works with one. 2 outliers. In order to make, the first one is alanine. I told you about the recognition of tRNA, where alanine is an exception on every respect. So alanine has no normal dimeric structure. This is one structure. You see the two active sites. This is another view of another organism. They are not connected. And there is no yellow. You don't see the yellow, of course, because that's a connecting, normally connecting. So you see the two active sites. That's one example, but they are different. And there are models, qualitative models. And this is the last one, the structure I showed you Tuesday. That's the structure. You see the tRNA, the amino acylation state. Here is the active site. In order to go to the editing site, it has to move, and the other subunit is there. So all the ingredients are like the class 2, they are there, but not in the same topological position. It's a bit bizarre. We don't know why. But you see, now that we have a structure, this is not a model. I mean, part of it is a model, but the starting point is the structure. And we know where it has to go, of course. We know these are the two active site and editing site. So the data are there. Question is open. Why do we have an oligomeric style different from this typical class? It's good because exception can lead us to some functional correlation and evolutionary correlation. I have no answer except the observation. For the other, it's more optimistic. I will just phenylalanine tRNA synthetase. That's the structure. You see the two active sites? I mean, it's one structure, but that's more conserved. This dimeric structure is conserved, and there are various But you see, it's not exactly like the other. What Brice found by analyzing the superposition, you see the two active sites, the yellow connecting is there. Here there is another active site, but with modification it's not functional. But this dimer is canonical in this dimer. In fact, what we have, it's two alpha structures. So these are canonical. These are canonical. So this entity, one of the subunits, in fact, it's a evolved dimer. So phenylalanine fits the canonical structure. So it enters the game. So we have only one exception left. And you see the rest, of course, the other group are different for other functions. I will stop there. I give the credit to Brice, I mentioned, who was helped me in assembling this comparison and in the end. And of course, I use most of the data from my own group, but they extend to all the things. And okay, I think now we have no time for the questions. Thank you.